A good day to everyone. I am Saranya Sree. As per the topic, I am going to present the strategy on how to develop drug to combat coronavirus. I am pretty sure everyone knows what coronavirus is all about, the symptoms, where is it originated from, but do you know how does it gain entry into our host cell? Well, it is very simple. The virus gains entry into our host cell by binding to a receptor on the cells called angiotensin converting enzyme type 2 and with the help of transmembrane serine protease, which helps to cleave the S protein of the virus, and thus the viral particles can actually fuse with the cell membrane. So my strategy will be by inhibiting the transmembrane serine protease type 2 enzyme. First step will be by creating the homology model of the serine protease. Firstly, we need one protein sequence and also one template sequence. The protein sequence will be obtained from in Fasta format from NCBI, and the template will be obtained from the protein database. So both of the sequence will be aligned together by using Swissmodel, and expected value similarities above than 30% is quite good enough, and there will be gaps between the similarities of the sequence, and that gaps will be filled with loop chains and side chains building by referring to protein database. And after that, the quality of the model will be evaluated by using molecular dynamic program, and then the interaction sites will be actually Second step is by preparing the potential inhibitors for serine protease, in which I have chosen based on my readings is chemostat mesylate, nafamostat, and also bromhexine hydrochloride. So all of these inhibitors structures are actually obtained from the protein database, and they are prepared for next molecular docking through the dock tools. Moving to molecular docking, we are going to dock all those 3 drugs into the active site of serine protease. The binding affinity will be calculated and confirmed by using scoring method in AutoDock simulation. Later on, the free binding energy, the type of interaction can be determined as well, and the drug with most compatible binding with the serine protease will be calculated, its inhibition constant, and selected. Now that we have chosen our candidate, the drug that we have chosen previously, now we are going to synthesize the drug, which is by through library synthesis. We are going to create a lot of similar compounds to that of the drug to increase and optimize the binding with our active site. And through continuous flow chemistry technique, we are going to obtain the percentage yield of activity between the drugs and the target site, and not to forget all the similar compounds that have been synthesized, that have been created actually, have to follow the Lipinski 5 rules. We have our lead compound, we are going to crystallize it through crystallization technique, which have 2 stages, nucleation and also crystal growth, and this is done to get our active pharmaceutical ingredient to determine its structure, its size, and bioavailability. And we are going to use controlled cooling technique, which will increase the purity and also the yield. Crystallization, we are going to determine the absorption, distribution, metabolism, excretion, and also the toxicity level of our drug compound by using the Asia flow chemistry electrochemistry system. Now we can move on to clinical trial. The phase 1 clinical trial usually have to do with in vivo and also with animal studies to determine the dosage and also toxicity of our drug. Later on, moving to phase 2, phase 3, phase 4, and then finally it will be to be approved by the FDA. That's all for drug development.